If there's no questions, let's get started. Let me share the screen. Are you guys hearing me okay?
Yep.
Thank you. Right here. So what we're going to cover today is chapter 4, and we should finish the chapter today. I think we're going to finish early, so I'm going to get started a little bit on chapter 5, but I'll let you go early because we're ahead of schedule. Any questions about that? There's no quiz this week. There will be a quiz next week. We have a long discussion that we'll have in the lab, and we'll talk about that. Oops, that's wrong. There's a long... I'm a little confused here. Have we done Lab 5? Okay, that says read the unknown instructions. I need to talk about the unknown project, and we'll be doing Lab 6 today. So I got the wrong lab up. All right. Any questions about what we're doing?
No.
Let's go ahead and get started then. I have this slide as the one we left off on. Did I talk about the Golgi apparatus or the Golgi complex? Both names are used, the Golgi apparatus and the Golgi complex, and sometimes they just call it the Golgi. No comments. All right.
I don't, yeah, you did.
I did talk about the Golgi?
Yeah, I think the last thing you talked about are the centrioles.
Okay. So it modifies proteins, and they come from the ER, and we'll talk about that in a minute, and then they're sent to various places in the cell. Let me state, I don't think I mentioned this, the Golgi apparatus, when you look at it over here, although the edges with the vesicles coming into it or going off of it, make it look different. It sort of looks like a stack of pancakes with maybe globs of butter. Any questions about that? And this is the artist's rendition of it. The Golgi complex, the endoplasmic reticulum, and the vesicles all together make up the endomembrane system. It's various eukaryotic membrane-bound organelles that are responsible for the cellular transport system, making a conveyor belt for molecules moving around in the cell. Especially proteins use the endomembrane system to be started, what do you call that, manufactured one place, modified another place, transported to where they'll end up at. And they use the endomembrane system for getting that to work. Let me see if I can get this. Oh, never mind. I have a link there, and I just put it there, but I can't run it from here. But I remember I've still got it up here. Let me get this out of the way. Let me know if you can't hear this well.
In this video, I'd like to talk about the endomembrane system. The endomembrane system consists of organelles that create, modify, and transport proteins and lipids throughout the cell. It consists of the nuclear membrane, rough and smooth ER, the Golgi apparatus, and different types of vesicles.
All right, oops, went a little too far. One of the words didn't come in well, so the endomembrane system includes the Golgi apparatus, the smooth ER, which, where is that? Does anyone see it? Maybe there. The rough ER right here, and the vesicles, which are sort of here and there, and maybe there. The little ones there, not the big ones, the little ones. Any question about that?
Vesicles. First, ribosomes, which are protein factories of the cell, can be found on the rough ER. These ribosomes create proteins and then transfer these proteins into the ER, where they undergo modifications, such as folding. These modified proteins may be absorbed into the membrane of the ER or packaged into a vesicle that may travel to the Golgi apparatus. The smooth ER may transport lipids to the Golgi apparatus, also using vesicles. A vesicle is a relatively small membrane-bound enclosed sac that stores or transports substances. At the Golgi apparatus, the proteins are sorted, packaged, and tagged so that they can travel to the right place inside or out of the cell. The receiving side of the Golgi apparatus is called the cis face. The opposite side is called the trans face. The transport vesicles that formed at the ER travel to the cis face, fuse with it, and empty their contents into the Golgi apparatus. As the proteins and lipids travel through the Golgi apparatus, they undergo further modifications that allow them to be sorted. The most frequent modification is the addition of short chains of sugar molecules. These newly modified proteins and lipids are then tagged. These tags help the proteins arrive at their proper destinations. Finally, the modified and tagged proteins are packaged into vesicles that leave the trans face of the Golgi apparatus. Some of these vesicles deposit their contents into other parts of the cell where they may be used, while others release their contents outside of the cell by diffusing into the cell membrane. So there we go, the endomembrane system. Thanks for watching, and Moo Moo Math uploads a new math and science video every day. Please subscribe and...
All right, any questions?
No.
All right. Let's come back. So the endomembrane system includes the endoplasmic reticulum, both the rough ER, which makes proteins, and the smooth ER, which makes lipids. And then they send molecules that they make to the Golgi apparatus using vesicles, and then the molecules are modified in the Golgi apparatus and tagged, and they come in one end of the Golgi apparatus, sorry, didn't move that, this end, and then they come out the other end, which has the bigger pancakes. And they come out of the Golgi apparatus once again in vesicles, and then move to where they're supposed to be in the cell, wherever that may be. That could be in the cytoplasm someplace, it could be in the cell membrane, and especially with proteins, they can also be deposited outside the cell. All right, any question about any of that? That is the endomembrane system. Now, lysosomes and vacuoles look similar to a vesicle. A vesicle is a membrane-bound organelle which is actively transporting things, we'll say molecules, from one location in the cell to another location. Okay? And that's just what a vesicle is defined as. Lysosome and vacuoles can start as a vesicle, but once they get to their final location, they then are given another name. A vacuole is a vesicle that is simply storing things, waiting for the cell to use them. So vacuoles are storage vesicles. And I'm not sure of this, but I think all vacuoles initially start life as a vesicle. And don't quote me on that because I'm not 100% certain that's right, but it certainly is right for the majority of vacuoles. It's not correct for the central vacuole, and that's a different story, but for other vacuoles it is more or less correct. And that is a vacuole started life as a vesicle. And then when it's no longer moving the molecules from one part in the cell to another, it's given the name as a vacuole, which is simply storing molecules for the cell. And that storage can be useful molecules like food, structural support molecules, or waste storage. In fact, in plants, the central vacuole stores many products that the cell wants to use, and then many products that the cell wants to store as waste. So think of it as your refrigerator, which you're storing all your food to keep it fresh, your steak, the milk, the bread, the butter, and then you start putting in your fluid waste from the toilet. Okay, that's what the central vacuole is in plants. And why they do that is a good question. It's just that plants, they just do that. And it's because they don't move around, so they store a lot of molecules, which animal cells don't tend to do that. Like their nitrogenous waste, they will excrete it from the cell. It'll be picked up by the blood. It'll be taken by the blood to the kidneys, and then excreted from the kidneys into the urine of the bladder, and then that'll be urinated out. That's what animals do. But plants, they store those wastes, and they store it within the central vacuole, where they have a lot of material they're storing for future use, like sugars or I don't know, various minerals they use, proteins that they made, and other things like that. Any questions about any of that? All right. Lysosomes look like a vesicle, but they have a specific function, and that is they tend to digest or lyse things. And they're called lysosomes because they typically have the enzyme lysozyme in them as one of the digestive enzymes. All right, any question about any of that? If not, let's move on to our next vesicles. We need to talk about our next, organelles. We need to talk about mitochondrion. Mitochondrion is singular, mitochondria is plural. It's the site of cellular respiration where the cell makes the majority of its ATP. So it's called the cellular powerhouse because ATP is the molecule that the cell uses most of the time when it needs energy. The mitochondria is unusual in that it has two membranes, an outer membrane shown here, and then an inner membrane which is greatly folded into cristae. And the belief is that the folding is to increase the surface area of the inner membrane. The mitochondria is unusual in that they contain DNA molecule inside it, and inside the inner membrane, inside this space here, we call that the matrix. That's not shown here. You should know that because we'll talk about that later in the term. Inside the matrix of the mitochondria, you can find a DNA molecule, and then ribosomes. And these ribosomes differ from other eukaryotic ribosomes in that they're 70S in size instead of 80S. Any question about that? All right, so that's the mitochondria. Let's now talk about the chloroplast. It is the site of photosynthesis, and so this only is found in photosynthetic cells, such as the green plant cells, meaning not the roots of the plant, but the green cells of the plant engaging in photosynthesis. In algae, which are also green, and then in other photosynthetic cells, which, like the red algae, wouldn't be green. The chloroplast is unusual in that it also has two membranes, an outer membrane shown here, and then an inner membrane which is highly folded and flattened. One of these is the granum, and you put them all together, the stack is the grana. And the point is it has a flattened membranous stacks that are called the thylakoids, and it's just part of the chloroplast inner membrane. Highly folded, I don't know what you want to call it, like the mitochondrion, except it's even more folded than the inner membrane of the mitochondria. And that's called the thylakoids. Inside the thylakoids, you can find a small DNA molecule and small ribosomes, also of the size 70S in size, smaller than the eukaryotic ribosomes of 80S. All right, any question about the chloroplast, the mitochondria, or any of the other organelles that we've discussed in this chapter? If there's no questions, let me open a new file here. And let's move on to the next topic. And that is the endosymbiotic theory. It is a theory to explain the evolution of the mitochondria and the chloroplast, which are only found in eukaryotic cells. How did these organelles evolve? And that's covered in your textbook, page 275 to 276, if you have the current edition of the Clark College version of the textbook. Hopefully everybody knows what I'm talking about when I say that, meaning the textbook that only has the chapters that we're covering. And that's on in chapter 10, page 275 to 276. I think the earlier versions of the textbook for Clark College, it's on page 266 to 68. All right, so the endosymbiotic theory states that a long time ago there was an ancestral prokaryotic cell that got incorporated inside of an early proto-eukaryotic cell. And it wasn't quite a eukaryote yet, because it didn't have all the characteristics of eukaryotic cells that we have today. But it was an early ancestor of the eukaryotic cell. And let me come on over here and go here. And so early, I'm just going to go euc for eukaryotic cell. So the early eukaryotic cell eats a prokaryotic cell. And for whatever reason, it did not immediately digest that cell, so it stored it. Inside its cytoplasm. I can't spell now. Cytoplasm. You know what I'm saying. I'm not probably spelling right, but that's all right. The eukaryotic cell, and we're going to call it a proto-eukaryotic cell because it wasn't truly a eukaryotic cell yet. The eukaryotic cell could make ATP from glycolysis, making 2 ATP per molecule of glucose. Have we talked about that yet? Glycolysis? I don't think we have. We'll talk about that, but the point is that the eukaryotic cell could make 2 ATP from a molecule of glucose. And how it could do that was using glycolysis. And we'll talk about glycolysis in a future lesson, I believe. And you can correct me if we've talked about it and I'm just flaking out and can't remember what we talked about. Okay? The prokaryotic cell inside of it, and let's go ahead and call it a bacteria because that's what it was. The bacteria inside could perform aerobic respiration making 38 ATP, and it could have been 36 to 38, we'll talk about that. We'll just say 38 ATP per molecule of glucose. And because the bacteria was inside of the proto-eukaryotic cell, the bacteria grabbed glucose from the cytoplasm. Okay? It was inside the cell, so it just grabbed glucose from the cytoplasm. And then it made 38 ATP. For whatever reason, the bacteria released some ATP to the eukaryotic cell. Probably what happened was back then life was very early and the ATP probably, we don't really know, probably was released by accident. I mean, it's possible the bacteria knew what it was doing. Of course, it's not a thinking creature, and realized, hey, let's make the eukaryotic cell like me, and then it'll keep me around and it won't eat me. And that's entirely possible. I have no idea. But anyways, the bacteria did release ATP, and the eukaryotic cell said, what? Oops, sorry. What happened? I lost 1 glucose and gained a whole bunch of ATP. And then you can say it argued with itself, I only can make 2 ATP per glucose. So the eukaryotic cell kept the bacteria inside of it, meaning it did not eat it. So it would make it, which is a little too many its, so it would make the eukaryote ATP. Any question about that? The eukaryotic cell kept the bacteria inside it, meaning it did not eat it, so that the bacteria would make the eukaryotic cell ATP. And let's change that it to bacteria. So the two became symbiotic, meaning they lived together, they lived in harmony with each other. The bacteria made ATP for the eukaryotic cell. The eukaryotic cell in return gave the bacteria a home and provided it with glucose. Any question about any of that? The two have now lived so long together in symbiosis that we no longer find the bacteria outside of a eukaryotic cell. And of course, almost all eukaryotic cells have bacteria inside them as mitochondria. So that's the theory. Any questions about the endosymbiotic theory in relationship to mitochondria? Any questions about it?
No.
Okay. Wrong one, sorry. What's odd is that not only did this happen one time, in plants it happened 2 times, meaning in plant cells, an early cell took in a bacteria which lived in symbiosis with it and became the mitochondria, and then later on the future plant cell took in a bacteria which became the chloroplast, and so this happened twice in plant cells, with one bacteria becoming the mitochondria and the other becoming the chloroplast. And the two live in symbiosis. The chloroplast bacteria, meaning that bacteria engaged in photosynthesis and made sugar for the plant cell. Any question about any of that? There's strong evidence for this theory. First of all, you can look at some protists and you can find that they take in bacteria and then live in symbiosis. This, what is that, paramecium, takes in a bacterium, and the bacteria can engage in photosynthesis and then make sugar for the paramecium. The paramecium gives a home for the bacteria and also gives nutrients for the bacteria so that the bacteria can make sugar. As well as will move towards the sunlight so that the bacterium inside the protozoan can then create more sugar. There's also amoeba, which I think I mentioned right here. Almost all eukaryotic cells have bacteria inside of them as mitochondria, and we have the mitochondria to make ATP, meaning the mitochondria make the majority of our ATP. But there are a few, not very many, but there are a few, actually I don't want to save that, I don't know what I'm doing. There are a few eukaryotes, just a few, what are they called? Amoebas. A few amoebas that do not have a mitochondria. And what these amoebas do is they find a bacteria, engulf them, and then start living in symbiosis with the bacteria. And the bacteria perform aerobic respiration, making ATP for the amoeba. So these amoeba are doing what we think the first eukaryotic cells did, meaning they engulfed a bacteria, and then that bacteria makes ATP from glucose using aerobic respiration. Any question about any of that? So there's some evidence, and that is we can actually see what we're proposing happening in a few cells. Another evidence of the thing is, well, let's talk about a weak evidence, and that is the cell, when it's engulfed by a larger cell, it'll have two membranes, its own membrane and the vesicle membrane of the phagosome. Do you remember what a phagosome is? That's when something is brought into the cell. Okay? And when we look at the chloroplast and the mitochondria, we see that these organelles have two membranes, an inner and outer membrane, which we would expect if at one time they were phagosomes. And these are the only membrane-bound organelles which have two membranes. Any question about that part? That's still pretty weak evidence, so let's look at some stronger evidence. The growth of the mitochondria and the chloroplast happens by binary fission. The growth of eukaryotic cells happens by mitosis. And of course, the growth of all prokaryotic cells happens by binary fission. If you look at the ribosomes of a eukaryotic cell, it's 80S in size. And if you look at the ribosomes of the mitochondria and the chloroplast, they're 70S in size. And the ribosomes of prokaryotic cells are 70S in size. Furthermore, if you look at the first amino acid which is made in eukaryotic cells, it's methionine. If you look at the first amino acid made in prokaryotic cells, it's a related amino acid, but not methionine. It's formylmethionine. And the first amino acid made in the mitochondria and the chloroplast is formylmethionine. Eukaryotic cells have their DNA wrapped around histones. Bacteria do not have their DNA wrapped around histones. The mitochondria and the chloroplast do not have their DNA wrapped around histones. Furthermore, you look at the DNA of eukaryotic cells, the chromosome is a linear chromosome, meaning the DNA molecule has a beginning and an end part. The DNA of prokaryotic cells is circular, meaning it wraps around and the ending part is where the beginning part is. And the DNA of the mitochondria and the chloroplast is circular. Furthermore, if you sequence the DNA of the mitochondria and the chloroplast, the sequence of that DNA does not fall within the domain eukaryotes. It falls within the domain bacteria, meaning the DNA of mitochondria and chloroplast is most closely related to the DNA sequence of bacteria. Any question about any of that? All right, that's the evidence for the endosymbiotic theory. It's pretty strong evidence that eukaryotic cells became eukaryotic cells in part by absorbing or eating a bacteria, which it then lived in symbiosis with to become the mitochondria. And in green plant cells and other photosynthetic cells, not only did they take in a bacteria which became the mitochondria, but they took in a bacteria which became the chloroplast. All right. Any questions about anything from Chapter 4? If not, that's it for Chapter 4. Let me save this down. And I will post this. Somebody should remind me to do that in the lab. Remind me if I haven't done it, because I've added a link to seeing, oops, went too far. The endomembrane system. The old link was no longer working, so I took directly a movie from YouTube and put it in there. I'm not sure why the old movie wasn't working, but it wasn't working, and so I removed it. And I hadn't put in a new movie until right before the lecture. So somebody please remind me to post this Chapter 4 slides up on the website and make them available to you so that you can view them. If I don't make them available for you to view them, somebody should remind me to do that, and then I will make them available for you to view them. Sometimes I forget. I think it was Chapter 3. I was several days late before I made it available for you guys to see them. And I intend to put them up as soon as we're done with the chapter. So Chapter 4 is ready to go up, except I've got to update the Chapter 4 slides because I changed this slide. Any questions about any of that?
Question. Is the test going to be on Chapter 3 and 4?
Let's take a look at the syllabus, because it doesn't lie. The next test is on Chapter 4 and the end of Chapter 3. Okay, as well as Labs 5 and 6. Chapter 5 will not be on the next quiz. All right?
You said that you were going to talk about the unknown project.
I'm going to talk about that in the lab.
Okay.
Okay? All right. I'll leave that up for the lab, although that's a long ways away. And let's go to chapter 5. We won't go long, I just want to get started on it. Let's see how many slides. 75 slides in this chapter. We won't do all of them today, obviously. Chapter 5 on microbial metabolism. The slide gives you the major goals of this chapter, not all of the goals. For all of the goals, you should look at the, what do I call that? The, not the chapter notes, but the chapter objectives. And a rough outline. So know the terms: catabolism, anabolism, activation energy, enzyme, active site, competitive inhibition, non-competitive inhibition, which is also called allosteric inhibition, redox reactions. You should know what a photoautotroph is, a photoheterotroph, a chemoautotroph, a chemoheterotroph, and you should be able to discuss, if I give you an organism, which of these it is, a photoautotroph or photoheterotroph, etc. You should know what fermentation is, and cyclic and non-cyclic photosynthesis. You should understand what feedback inhibition is, and then we'll talk in great detail about what aerobic respiration is. You should understand it, know its steps, and how it differs from anaerobic respiration. Any question about what we're doing? All right. So this is where we'll talk about aerobic respiration, which happens in the mitochondria, which I just talked about that. Only I said, did we talk about that or not? I didn't remember if we were doing it. It gets all blurred up because every term I'm teaching this and I get confused from one term to another. And generally it's about the same. Actually, there's 2 students in here who have taken my class before. So is this about the same? Let's see if they're on here. Neither of them are on. All right. Let's talk about metabolism. Metabolism is all of the chemical reactions occurring inside the cells of an organism. And if for a prokaryotic cell, which we're talking about the bacteria mainly here, metabolism then is all of the chemical reactions inside the cell, because bacteria only have one cell for the organism. You can break metabolism down into 2 parts, catabolism and anabolism. Catabolism is the decomposition reactions where you're taking a complex large molecule and breaking it down into a smaller molecule. Energy will be released. And then anabolism is the reverse, taking the smaller molecules and making them into the bigger molecules, the more complex molecules. The anabolic reactions or biosynthesis reactions, meaning we're making larger molecules, and they take the energy coming into the system for these reactions to go, meaning they take in energy, and then the catabolism reactions give off energy. A bigger molecule contains more energy in the chemical bonds than smaller molecules. Whenever you change energy from one source to another, including from a complex molecule to a simple molecule, some of that energy will be released as heat, and this is a law of the second law of dynamics, and that is whenever you change energy from one form to another, some energy is lost as heat. And that's true also when you're changing the energy in the simple molecules to a larger molecule. Some of that energy is lost as heat. Heat is just another form of energy, and we say it's lost because heat isn't terribly useful to the universe. Heat, you can't do anything great with heat. You can't use it to make a larger molecule, meaning the cell can't use heat for its chemical metabolism. Okay, any questions about any of that? And if you've had biology, you know that warm-blooded animals do use the heat given off by mainly catabolic reactions, although anabolic reactions also release heat. They can use that heat to keep the animal warm. So in some ways, we can use that heat a little bit. Okay? Let's see. Catabolism reactions release energy. Sometimes that energy can be released and used to make ATP. But this slide is a little bit of a misnomer, because in animal cells there are only two chemical reactions that can happen where the energy from a complex molecule being switched to a simpler molecule can be used to generate ATP, meaning of all the chemical reactions going on in your cell, and there are thousands of them, only two of them can be used to generate ATP. Now, animal cells are not very good at generating ATP, and I'll go ahead and tell you, we'll talk about both of those ways in this lesson. One is glycolysis, which I talked about in the last lesson on chapter 4, and the other is aerobic respiration. And we'll talk a little bit more about those two reactions in this lesson, but not today. Okay, so like I said, this reaction is a little bit, or this slide is a little bit of a misnomer, and that is the majority of the molecules being broken down into simpler molecules do not generate ATP in animal cells. Even in plant cells, the majority of the complex molecules being broken down into simpler molecules do not generate ATP, although green plants are much better at generating ATP than animal cells, so they have more than two reactions that can generate ATP. But the majority of the reactions do not generate ATP. Okay, everyone clear on that? The majority of complex molecules, when they're broken down, do not generate ATP. On the other hand, when single molecules are taken to make complex molecules, such as glucose being taken to make starch, ATP is used up because you need energy to go from a simple molecule to a complex molecule. So all anabolic reactions, I shouldn't say all, almost all anabolic reactions require energy from ATP. And the ones that don't take it from ATP take the energy from some other molecule. So all of them require the energy from some molecule. An example of a molecule in your cells which, when it's broken down, does not generate ATP is when you take sucrose, that's simple table sugar, and break it down into glucose and fructose. The cell has no way of making ATP out of that. Any question there? The only way you can make ATP is from glycolysis or from aerobic respiration. All other chemical reactions that you do in your cell that are catabolic do not generate ATP. All right. Let's briefly talk about metabolic pathways. A chemical reaction is when you start, let me blow this up, is when you start with a molecule, A, and it's changed, usually by an enzyme, into molecule B. That's a chemical reaction. But you could take molecule B and convert it into molecule C by another enzyme, and molecule C can be converted into molecule D by another enzyme. Molecule D can be converted into another molecule, molecule E, by a fourth enzyme, and then molecule E can be converted into molecule F by a fifth enzyme. And then when you string them all together, this is a metabolic pathway going from molecule A through a bunch of intermediate products to the end product, meaning the last molecule made, molecule F. And this is what a metabolic pathway is. Metabolic pathways are determined by genes because genes encode the enzymes that are required to convert molecule A to molecule B. A second gene makes another enzyme converting molecule B to molecule C, and then an enzyme making molecule C to molecule D to E, and then E to F. F is called the end product because it's the last molecule made. A is either called the starting substrate or the starting reactant, because that's where we start. And all of the other molecules, rather than calling it the end product of enzyme 1, we call it the intermediate product. All of these others. Okay, any question about that? Enzymes speed up chemical reactions. I think we'll talk about that in this lesson, but probably not today. And enzymes convert one molecule to another. Any question about any of that? All right, here is all of the, guess I shouldn't have stopped the slideshow. All of the chemical reactions that we know of happening in one of your cells. Obviously, there's probably many that we don't know of, but here are all of the chemical reactions happening in your cells. And you do need to know all of them. Only kidding. There's a huge amount of metabolism. That's just all that slide was trying to show you. And all of that metabolism happens in your cells because of enzymes catalyzing a chemical reaction. Any question about any of that? All right, no question. Let's move on. Chemical reactions happen because of the collision theory. The collision theory states that chemical reactions occur when atoms, ions, or molecules come together and collide. If they have enough energy in their collision, then the chemical reaction will proceed. The activation energy is the energy needed to disrupt the electronic configuration in the molecules, changing the reactant or starting substrate to the next molecule, which we'll call the product of that chemical reaction. Any question about any of that? The reaction rate is the frequency of collisions with enough energy to bring about a reaction, meaning where one molecule is changed into another molecule. The reaction rate can be increased by increasing the abundance of enzymes. The more enzymes you have, the more of the starting product or starting reactant will be changed into that product. And you can also increase the reaction rate by increasing the temperature. By increasing the temperature, you give the molecules higher energy. They're moving around more rapidly. And if they have higher energy and they're moving more rapidly, when they collide, they're more likely to have the energy they need to reach the activation energy. You can also increase the reaction rate by increasing the pressure, and this one you don't need to fully understand, but if you increase the pressure, you make the activation energy easier to come to because of the fact that when you increase the pressure, you put the molecules closer together, and so they're more likely to collide. Okay? And like I said, you don't need to understand that, but increasing the temperature or increasing the pressure increases the reaction rate. Here is a chemical reaction, and we're seeing the reactants on the y-axis have a certain level of energy, which is why we're putting them there, and the products have a lower level of energy, why it's lower on the y-axis. On the x-axis, they're just showing you time. It's not stated. In fact, the y-axis really isn't stated either. But the reactants here can be converted into the products, and when the reactants come together and collide, if they have enough energy, they'll be converted into the products. And you can think of this being something like glucose being converted in a chemical reaction to glucose and carbon dioxide and water. They have fewer energy. Carbon dioxide and water has fewer energy than glucose. So when glucose collides with oxygen, it can create carbon dioxide and water. And when the molecules collide, they have to have this amount of energy. Up here, the reaction without an enzyme, before the molecules will have enough energy to change the reactant into the products. And that amount of energy, from what the reactants have to how high they have to be without the enzyme, is called the activation energy without an enzyme. For glucose, I could convert glucose into CO2 and water simply by taking a blowtorch to the glucose and then burning it, giving it enough energy to reach the activation energy to create carbon dioxide and water. Any questions about any of that? All right. Now, obviously, a blowtorch is really hot. I don't know how hot it is, but it's really, really hot. You certainly don't want to ever touch a blowtorch, okay? I'm talking about the flame part of it. And yet, glucose being converted to CO2 and water happens all the time in the room you're in. Where is glucose being converted to CO2 and water and not requiring the energy of a blowtorch? In your room. Where is that happening? Come on, we're going to stay here until somebody answers the question. In your room that you're in, where is glucose being converted to CO2 and water, and it's not requiring a blowtorch amount of energy, the activation energy without an enzyme?
Breathing?
Say again?
Breathing?
Close. What you're saying is it's happening inside you, and breathing is required for the glucose to be converted into CO2 and water. But what I'm trying to say is it's happening inside your cells. Glucose is being converted from glucose to CO2 and water, and that is not requiring the temperature of a blowtorch, the activation energy without an enzyme. And the reason is because inside your cells you have enzymes which allow this reaction to happen at a much lower temperature than a blowtorch. And the enzymes in your cells can convert glucose into CO2 and water. And what temperature are your cells? Come on. What temperature are your cells?
98.6.
98.6, thank you, or 37 degrees Celsius. That 98.6 is Fahrenheit. So obviously this is a much lower temperature, the activation energy with an enzyme, than the activation energy without an enzyme. Everyone clear on that? And this scale isn't shown to exactly scale, meaning this is just a very slight energy hump, and this is a tremendous energy hump. All right, enzymes are biological catalysts. Catalysts are things that speed up chemical reactions, where they convert a reactant to a product. Okay, any question about that? So enzymes are simply biological catalysts. They speed up chemical reactions. And you don't need to know the numbers here, but they speed up biochemical reactions by a very large amount, 10 to the 8th to 10 to the 10th times the normal reaction rate. Enzymes are encoded by genes. So in the DNA, you have a coding for all of the enzymes that your body needs. Enzymes are, for the most part, specific for a single chemical reaction. They bind specifically to a single specific substrate. The specificity of the enzyme to the substrate is because of the three-dimensional folding pattern of the enzyme. Specifically has a region called the active site, we'll talk about it in a little bit, that's in a specific shape that binds to the substrate. And generally speaking, an enzyme will only bind to one substrate. Now that's not entirely true. Those of you who have worked with drugs know that we have some drugs that mimic a substrate. And so you can take this drug and instead of the enzyme binding to the substrate and converting it to the product, the enzyme will bind to the drug, and then the enzyme no longer makes the product, and that's why the drug works. Meaning, it's not entirely true, but for this class we can say that as a rule of thumb, enzymes bind to one specific molecule. And in the case when they don't bind to one specific molecule, like they bind to one molecule and a drug, the drug is mimicking the shape of the substrate. Okay? Any question about that part? And then lastly, enzymes are not used up and altered in a chemical reaction. So when a biological enzyme grabs onto a substrate and converts that substrate to a product, at the end there, that enzyme returns to its original shape and then can grab another substrate and then can repeat this chemical reaction countless numbers of times. The enzyme is not used up or altered in the chemical reaction. That doesn't mean that the enzyme isn't changed. While the enzyme is acting, the enzyme actually binds to the substrate. And we're talking about a covalent bond, usually, although it could be a hydrogen bond. Usually it's a covalent bond. It forms a complex with the substrate. And that complex is what's putting the stresses on the chemical bonds of the substrate to change it into the product. So the enzyme substrate complex orients the substrate to increase the probability of the reaction occurring, lowering the activation energy of the reaction. All right, any question about any of that? If not, let me go ahead and just talk about this slide here, and then we'll stop. Enzymes normally end with ASE, ACE. Most enzymes have ACE on their name. And here are a few examples. You don't need to know these examples. We'll talk about ligase in the lab. Transferase, we'll talk about that in this lesson. Oxireductase, I don't think we talked about that. But catalase is another enzyme we'll talk about in the lab. Most enzymes end in ACE. There are a few that do not, like if you know the enzyme renin, the enzymes that were named a long time ago, such as renin, they were named before the terminology ACE came around. But most enzymes do end in ACE. Any questions about any of that? If not, we'll stop here 10 minutes early, and I'll see you in the lab at 6:30. Are there any questions? If there's no questions, I'm going to end this. All right, see you in the lab.